Welcome back to the Imprint Hub, where we talk about maternal and pediatric pharmacology. Today, we're taking a closer look at developmental pharmacology in a particularly challenging environment, which is using external life support. When babies and children need these machines, every medication decision becomes more complex because now drugs are interacting not only with a developing body, but also with an external system. In this episode, Dr. Aislinn Pruss joins us to explain how extracorporeal systems change drug levels and how clinicians and modelers work to keep these patients safe and effectively treated. This video can absolutely stand on its own, but if you'd like to go deeper into the basics of developmental drug modeling and Dr. Pruss's research, check out part 1 of this conversation, linked in the box at the top of the screen or in the description below. So to build on our discussion about treating very young and medically fragile patients, can you talk a bit about how extracorporeal life support comes into play in your research?
In all patients that were under 2 years old, this drug that we were studying, which hopefully we'll publish about it very soon, we were looking at patients that it's typically used in very sick patients that are undergoing surgery or that are in the ICU. And the only data that we could find that was useful for our model in patients under 2 years was patients that had to undergo something called cardiopulmonary bypass. And this is also known as extracorporeal membrane oxidation, also known as ECMO. And it's basically where you're operating in or around the heart. And so you need to provide an alternative method of oxygenation for the blood so that you can keep your patient oxygenated and actually alive while you're maybe, operating on that very delicate organ that is supposed to be doing that for you.
Let's talk a little bit about the knowledge gap when it comes to developmental pharmacology and extracorporeal life support, because it sounds like it hadn't initially been something that you had considered in your research.
Yeah. So at first, it wasn't really something that we thought about at all. But once I noticed how different our pediatric models were when we applied them to these populations, because when we did have populations over the age of 2 that were not on ECMO, that were just undergoing like a normal surgery, and they had really good sampling and they had really good dosing that we were able to replicate in our model. And we were able to fit really well to those individuals. But once we got into these other populations that were undergoing ECMO, our volumes of distribution were much higher, like in prediction, than they were in actuality. So we were highly over predicting our volume of distribution, and we were over predicting our clearance as well. So we were expecting our patients to have a much higher concentration of drug in their plasma, and we were expecting them to be eliminating that concentration much faster than they were. And eventually I kind of pinpointed that the common theme in all of these things was the ECMO and the cardiopulmonary bypass. So I just Googled, does ECMO or cardiopulmonary bypass, does that machinery have an impact on drugs? Does it interact with drugs? And then immediately I came up with multiple papers where they ran in vitro as well as ex vivo studies on different drugs through this machinery. And they found that not only does the machinery pull out blood, so it's going to pull out all of the drug that's also in the blood because it's taking all of the blood, right? It's filtering it all through. But as it filters it through, that drug, specifically more drugs than others, depending on their protein binding, depending on their pH, depending on their composition, they can adsorb to the tubing that is involved in that. So some of that drug is going out and it's going into that tubing, and then it's staying there, and it's not going back into the body. So that's a form of clearance right there, which is really important. And it's something that I found in the SimCip workspace when I looked at all of the additional organs that I was able to copy and incorporate. None of them had their own clearance in there. So I had to also go and include an additional clearance in the clearance tab that was not related to the organ tab, which made it a little bit more complicated. But it was really exciting and it was really cool to see. And I basically found papers saying that anywhere between like 72 and like 90, like over 90% of the drug that we were studying could get stuck in that machinery, in that tubing, and could not be returned to the body. And once I accounted for it, was very significant. And I mean, that has a huge impact on like our pediatric patients in general, because you're having to give them so much more of the drug than you would expect. And then if you take that data, like if, for example, you're operating on a patient like that and you give them a bunch more of the drug, like 90% more, because 90% of it is going into a machine, you're going to say, okay, the next time we operate on a child, we should have this as our target dose. But let's say that that patient isn't undergoing ECMO, you could be severely overdosing that patient because they're not having that additional clearance from ECMO. So I think that that is something that's really exciting and really, really important for us to be studying. And I would love to see a study on the pharmacodynamic impacts of that as well, whether or not that's impacting their ability to be sedated properly, how it's impacting their outcomes. But I could digress on that one for a while. Anyways, so I incorporated the anticipated volume of distribution from that. And what I did is I took a copy of the heart. So SimSip has like default parameters and like default libraries for each organ, which is amazing. Um, and I looked at both the heart and the lung because I figured that it could kind of be duplicative of both of them, since it kind of acts as an oxygenator and kind of a pump back into the body. I found that the heart was the better option and that it fit the model better. Um, and I tailored the percentage of blood flow in the body that was diverted to it, as well as the expected percentage of body weight that the organ was. So you're kind of estimating like the size of your organ and how much of the blood it's diverting and taking away from the body. So how much of your drug is it going to pull away from your body? And then in the clearance, you also estimate how much of the drug is it not going to give back to your body. And that accounted for our overestimation of our volume, because our drug volume of distribution, it was getting taken out, right? So that immediately lowers it down to where our observed data was. So that validates what we're thinking, that this drug is getting taken out. And then with the clearance, that drug is slowly getting pulled out and more of it is getting stuck in that circuit each time. And a lot of it isn't getting back to the liver because your body's not oxygenating the way that it really should be, like your body's not able to process it as well. So that also accounts for the slowness, the very slow clearance that you experience with those patients. Our model worked super well in adults, right? So then we took that same model and we scaled it down to pediatrics. And so this is a patient that is between 6 months old and 2 years old. And the red line is where we did not add any sort of extra systemic clearance to account for that ECMO model. And we also did not include ontogeny of clearance. So the differences in that ECMO model based on age, which is also really important. And then this purple line right here, as you can see, there's a much higher rate of clearance than there was in our observed data, which is these blue dots right here. You can see that we anticipated that there would be a much, much, much higher rate of clearance in these patients when there really, really wasn't. And that is because with that ECMO model, you've got the blood leaving your patient's body and going into a machine, getting run through that machine, and then getting put back into the body. That slows down your clearance very significantly because you can't break it down if it's not in your body, which makes it a little difficult. The other issue is that for patients of different ages, they also are expected to have different heart rates and different rates of oxygenation and different flow rates of their blood through their body. So with that ECMO machinery, before we accounted for it, we were expecting that our patient's clearance, even with that ontogeny model, would be much faster than it actually ended up being. We also highly overestimated the volume of distribution, so just the total amount of drug that would be present. And so once we incorporated that machinery as an extra organ, and then we also gave it its own ontogeny because of the differences between heart rate, blood flow rates, and distribution of blood in the tissues between, neonates and infants and older infants and children, we were able to much better predict what the flow of the drug through the body would look like. And so this green line right here predicts what we estimated that the average patient between 0 and 2 years would look like. And after a lot of troubleshooting and a lot of ontogeny equations, most of which was actually just like fitting. So, really just looking at what the data that we had was and testing a lot of numbers and running a lot of simulations and then running simulations of simulations to figure out what the best fit was. We were able to get that best fit. But this describes really the importance of as individuals age, they're going to have not only different clearances within their body, but if they're having any sort of extra procedure done or if they're working with some sort of medical device, that medical device could be tailored to their age or to their size. And then you also have to account for that too. So that's a form of almost like ex vivo ontogeny, which was really, really cool that we were able to incorporate.
Thank you so much for joining us, Dr. Pruss, and thanks to you, our audience, for being here. The rest of our conversation with Dr. Pruss, where we get into working with the data that you have, genotypes, and bringing modeling into the real world, should be available soon. So we look forward to seeing you there. Let us know in the comments below if there are any topics that you'd like to see us cover, and we'll see you next time.